Today we will discuss a new term, that is UFMylation, and its role in regulating cell fate. The new term, by new term I means that UFMylation, we will discuss this UFMylation first time in our channel. That's why I called it as a new term. So this UFMylation is related to the ER stress, and that we have covered many times in our previous videos. So if if you have understood about this ER stress, then it will be uh, better. Then it will be better, or you can say that you will understand this upmylations topic in a better manner. So whenever there is a ER stress, so it will trigger different signaling pathways, and that helps in controlling the ER stress. Or otherwise, if the ER stress is not been controlled by those pathways, then it might trigger apoptosis. So those two pathways are this IRE1 alpha and PERK alpha, and both of them will cause the subsequent upregulations. There are one more pathways, that is ATF6, that is not shown here. So let us just focus, let us keep our focus on these two pathways only, that is IRE1 alpha and PERK, for understanding this mylation. So this ER stress will trigger this IRE1 alpha and PERK pathways, and that will lead to the upregulation of this XBP1 and ATF4. And XBP1 is in the spliced form, that is intron will be removed whenever the ER stress is there. So those XBP1s will uh, leads to the transcription of this UFM1, and that will cause, or that will help in preventing the ER stress, and that can prevent the ER stress by down-regulating this IRE1 alpha and PERK alpha pathways. So this is just a brief overview of the role of UFMylations in this ER stress. And if this ER stress is not been controlled by this IRE1 alpha or PERK pathways, or this UFM1 pathways, then it will trigger the ER dependent apoptosis. So now let us go through some of the examples. So this example is about the B cell differentiation. So in this B cell differentiation, this LPS will trigger this ER expansion, and during this LPS will trigger this ER stress, that will lead to the upregulation of this IRE1 alpha and PERK pathways, and it will cause this increase in the XBP1, spliced form of XBP1, and that will cause the production of this UFBP1 transcript. And this UFBP1 transcript in other way will cause the production of this UFBP1 or the UFMylations, and that will suppress IRE1 and PERK pathways. So you can consider this as a cycle process, that is, when the ER stress is there, then the XBP1 will produce, it will cause this UFBP1 productions, and then it will suppress IRE1. So in this cycle process, what will happen is it will allow this ER to get expand during this cycle process of this ER stress and ER stress induction and ER stress suppressions. And this expansion of this ER will cause this B cells differentiation, or this cells differentiation. So this is one of the examples that are been observed by in the B cell differentiations. And now this panel C is about the breast cancer, how this UFMylation works in the breast cancer. In the breast cancer, this E2 will we can say that the cytokines, when it entered the cells, it interact with this ASC1, and then this will help in the formation of this complex, that is ER alpha ASC1, and this complex formation will help in the poly UFM1 formations, and this poly UFM1 formulations will then change the transcription pathways, or will then will then leads to the epigenetic changes in such a way that this AREC1 ER alpha will interact with this P300 and SRC complex, and this P300 and SRC complex has a role towards the cell proliferations and the upregulations, and this complex formation happens in a way that it will upregulates the proteins upregulates the proteins that helps in this cell proliferations. So in this case what will happen is that this UFMylations helps in the cell proliferations, whereas in the B cells what the UFMylation does is it helps in the cell differentiation. And in the A case what does it is UFMylations tries to prevent this ER dependent apoptosis, or we can say that if the ER stress goes beyond the threshold level, then it can trigger apoptosis. So these are the three examples by which these UFMylations can regulate the cell fate, and these things are related to the ER stress. And why this ER stress is created, you can go through our previous videos to understand the ER stress topic in detail, and you can find those in the I button. Go find those videos link in the I button. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.